I got a 44 in IB and I've been tutoring it for 4 years, and I can guarantee you that 95% of students will miss this. This is the specimen paper that IB has released for IB Bio 2025. As you'll be aware, there's no past papers for the new biology syllabus, and therefore this is the only official IB resource that resembles a past paper. Today we're going to be looking at the exercises for theme A, and then in further videos we'll look at B, C, and D, as well as the ones for higher level. So let's get started. We're going to first look at the standard level paper 1A. This is similar to the paper 1 in the previous syllabus, so it's just a multiple choice. Let's start with the first question, which is what is a consequence of the ability of water to form many intermolecular hydrogen bonds? For each question, I'll do a counter of 3, and you can pause there and think about it, and then I'll talk through the answer. So in 1, 2, and 3, the answer is C. Why? Well, for A, ice does have a higher density than water, and it does float. But that is not because of intermolecular hydrogen bonds. That is because when water freezes, the bonds pack together more. The molecules can pack more, and that means that ice becomes more dense. The second one, light can pass through water, is also true, and algae can photosynthesize, but that is not because of the ability to form intermolecular hydrogen bonds. That is a physical property of water. In terms of D, small amounts of energy are needed to change water from one state to the other. That is not true. It's the opposite. So a lot of energy is needed to change water from one state to another. And then finally, C is true. Small invertebrates can walk on water surfaces, such as some mosquitoes, and that is because of the intermolecular hydrogen bonds, which form a surface tension in the water, meaning very light animals can walk on it. So hopefully that makes sense. On to the next question. What functions of life are being shown in these images? Now, very important to stick to what the question is asking. This is a vital thing for any IB paper. Read the question carefully. It's not what functions does organism X do, but which ones are being shown. Now, look at the image, and again in 1, 2, and 3. Question, which is a feature of phloem sieve tube cells? Now, you'll hopefully remember this from the cell theory. Again, 1, 2, and 3. The answer is B. Now, they have no nucleus, and that's an exception to the cell theory. Remember, these cells form part of plants. It's where the water travels up, and they don't have numerous chloroplasts. They also don't have lignified walls, and they do have a cytoplasm, but they do not have a nucleus, and that makes them an exception to the cell theory. So that's just something you need to remember, unfortunately. The next question is, what can be identified in the electron micrograph? Now, IB is known for having really bad quality images in their papers, and there's no exception, but just make sure that you look at the most distinctive features and play the game of discarding the ones that are very obviously not true. If you do that, you'll get to the answer fairly quickly. So 1, 2, and 3. So the way I would answer this question is, firstly, I would realize that this is not a prokaryote. Why? Because the prokaryote, first of all, has no organelles, and this cell clearly has organelles. You can see them here, right? These are compartments inside the cell itself, so that cannot be a prokaryote. In terms of an animal cell, it can also not be an animal cell. Why? Because these are chloroplasts. Now, you might not be able to identify that, especially if you just start IB, but it'll become obvious once you look at many more cells that this is a chloroplast. So it cannot be an animal cell. And then a plant cell, you have to read the answers, but B makes no sense. Why? Because it says large central vacuoles containing nuclei. Now, vacuoles are an organelle themselves. They don't contain a nucleus. The cells contain a nucleus, right? So B just makes no sense. Therefore, the answer is A. It's a plant cell with cell walls that you can see here, nuclei, a nucleus that you can see here, and then chloroplasts, and that's what you can see here. So hopefully that's all good. The next question is, what can lead to the emergence of analogous structures? Now, you need to remember what an analogous structure is for this, unfortunately, but hopefully you do. And 1, 2, and 3. The answer is B. Why? Well, C and D make no sense. I definitely recommend, when looking at these questions and practicing, try and discard the ones that don't make sense first. That way, even if you're not sure of the answer, you can make an educated guess. So C and D are referring to a niche. That's ecosystems, that's ecology. That has nothing to do with evolution and analogous structures, so they don't make any sense. And then in terms of A and B, which one is it? Well, it's B. Why? Because analogous structures, remember, are structures that have the same function, but they are in organisms with a different evolutionary past. Whereas A, so divergent evolution from a recent common ancestor, that's homologous structures. So just remember those definitions, right? It's the opposite. Homologous structures are similar structures, but they don't have the same function. Analogous have the same function, but they come from a different common ancestor. So they don't have a common ancestor, sorry. So that's all for the multiple choice in the specimen paper. Now let's look at paper 1B. So that's shorter answer questions. So first of all, suggest a reason for using a lower power objective lens. Now this is a very practical question. Hopefully you've used a microscope and you know the answer. But it's 1 mark. Whenever it's 1 mark, the number of marks indicates the number of ideas, bullet points, / concepts that you have to write down. But I definitely recommend doing 1 or 2 extra if you can and you have time, because you never know. If one of them fails, you'll have a backup. So again, 1, 2, and 3. And here are the answers. So first of all, well, answer 1 and 3, in my opinion, is basically the same thing, but IB decided they're different in their mark scheme. So it's basically being able to find the specimen or the interesting part that you're looking at in the specimen. So when you use a lower power lens, you get a more wider view of the slide, so that you can choose what you want to zoom into. And then the second one is reduced risk of cracking. Now, you might not have thought of this one, but it does make sense. Why? Because a lower power lens is smaller. So it's not as big, and therefore there's a lower risk of it actually touching and cracking the slide when you look at it. You might not have thought about this, but definitely write this down in your notes now, so that if a similar question comes up again, you can write it down. Sometimes the mark scheme, I agree, it's very difficult to predict. They will put things that are just very random, but you have to practice so much that by the end you basically know what they're expecting of you. So now, the next question, there's going to be tons of questions on this one image. So the images show examples of Poecilia reticulata. So take a while to look at it, study it, and then the first question is state the name of the genus to which it belongs. Now, this is super simple, actually, if you know what a genus is. 1, 2, and 3, the answer is Poecilia. So reticulata is the species name, and Poecilia is the genus. Remember, this is the hierarchy of taxa, right? And that's just the way it works. You need to remember that, right? Genus and species name. The next question is outline Linnaeus's morphological concept of species. Remember, this is not the current concept of species, but the one by Linnaeus. So again, take your time to think. And 1, 2, and 3. So the Linnaeus concept is that a species was a group of organisms with visible shared traits. So it was based on physical characteristics, right? Appearances. Why? Because back then they didn't have the tools to actually look at the genetic material, for example. So this is all they had. But again, this is not what we look at nowadays when we look at what species an organism is in. So now the next question, this is kind of a weird one. So 2 alternative hypotheses were suggested. The guppies were from a single interbreeding population, or the guppies were not from a single interbreeding population. And it asks you to explain which hypothesis is stronger. Now, there's not a single correct answer to this, so hopefully that's a hint. Think about it and see what you come up with. It's only 1 mark, so you only need 1 concept there. So 1, 2, and 3. The answer is it can be both. You can say that they're probably from the same interbreeding population, so the same species, because they have a similar body morphology. Or you can say that they're from different populations because they have variety in pattern and color, which is the same thing. You can just say that they have different body morphology. So both things go, right? As long as you state it, but you need to take a stance. You can't say both. You either say that you think they're from the same one or from a different one, and you explained why. And you'll get the mark. So this is quite an easy question, actually. It's just a weird one to answer. The next one, again with the image, is suggest how additional evidence should be used to further test which hypothesis is supported. Now, this is where you have to remember the current definition of species and what we do to classify organisms into different species. So take a second to think, and in 1, 2, and 3. So remember, we define species as being able to interbreed with one another, right? The organisms need to be able to interbreed and produce fertile offspring. So you could look at the mating behavior, so look at whether they mate or not. Best, I think, is to do DNA analysis, as it's suggesting. That's the most sure one, the most secure one. And thirdly is to gather information about reproductive isolation, which again is basically the same as the first, but reworded, right? So look at whether they can reproduce or not. Great. So the next and final question on this is to suggest an adaptive advantage for their striking color patterns. This might require more thinking, but it's 1 mark, so as long as you have one idea, you're probably good. So think about it, and 1, 2, and 3. So it could either be sexual selection. Remember, birds, for example, sometimes have different mating dances or feather patterns to mate. Well, it could be the same with the patterns of the fish. Or it also could be camouflage, right, to protect them from predators. So again, those are 2 valid ideas that I think are quite straightforward, so I won't really go into explaining them. So now we're on to paper 2. So remember, paper 2 now is also divided into 2A and 2B. 2A is technical questions, a shorter answer, and then B is those longer essay-style questions. So for paper 2A, the first question is state 2 membrane-bound organelles that are common to both plant and animal cells. Now this should be easy because there's a ton. So 3, 2, and 1. All of these, right? The nucleus, the mitochondria, the Golgi, the rough ER, the smooth ER, the vacuole, the lysosome. Basically, all of them except for the chloroplast, which is only in plant cells. It's exclusive to those. It's not found in animal cells. So the next one is 1 organelle that's found in a plant cell and not an animal cell. So I completely spoiled that, didn't I? But the answer is chloroplasts. That's the only one that's exclusive to plant cells. And then the third question is 1 organelle that is common to prokaryotes and eukaryotes. So this might be a bit trickier, but think about it. And 1, 2, and 3. The answer is ribosomes and the plasma membrane. Ribosomes are required in both because they help create proteins, remember, and then the plasma membrane makes a cell, right? Because otherwise it wouldn't be a cell, it would just be part of the medium. So it allows to contain the cell within itself. So that's enough for organelles. The next question is on DNA. So the diagram shows a stretch of DNA with complementary base pairing. And the question is asking to identify 2 features of DNA that distinguish it from RNA, and they are shown in the diagram, so it's quite simple. One of them is very obvious, I think. Think about it, and the answer is, in this case, 1, 2, and 3. Firstly, that DNA is double-stranded, whereas RNA is single-stranded. That might be less obvious, but then the second one, hopefully you got to that one, it's that DNA has thymine and RNA has uracil. Remember the other bases, adenine, guanine, and cytosine are shared, but thymine is the only one that in RNA is substituted by uracil. And then RNA is never double-stranded, so that's also important, but DNA is always double-stranded. Perfect. So now explain the significance of complementary base pairing. Now I think this one's a bit more tricky actually, but you might be able to come up with some answers. I think it's very important to know that in IB, even though in this case it's asking for the significance, it's always good to explain what complementary base pairing is, right? Always give some more information that you think is necessary. You might think that they would be very stringent when marking, but it's not the case. In the mark scheme, there's always extra points for just writing some facts that are associated with the question. So 1, 2, and 3. Well, again, as I said, so you get a mark for saying that adenine pairs with thymine and cytosine pairs with guanine, which is explaining, right, what complementary base pairing is. And then in terms of its significance, well, firstly, it ensures continuity of genetic information from replication. So this is very important. If you think about it, if we were to replicate DNA and then adenine, instead of pairing with thymine, could pair to guanine or cytosine as well, then what would be the point, right? There would be no genetic information. You would just get new genetic information every time you copy DNA, and you don't want that. It would lead to cancer, malformities, etc., etc. Second of all, it allows for stable arrangement of anti-parallel strands. So it's very important that DNA is very, very stable, right? It needs to be there for years. And this is helped by the fact that cytosine and guanine have 3 hydrogen bonds, adenine and thymine bind by 2 hydrogen bonds, and that maintains a stable, very, very stable structure, much more stable than RNA, for example. So those 2 strands really help a lot. And then thirdly, or fourthly, I guess, it provides a guide for DNA transcription. So it's similar to the second question, right? It ensures that the genetic information is passed on accurately. If you were to transcribe DNA and then adenine, instead of getting binding to thymine, binds to cytosine, you would get proteins to be completely different every single time you transcribe DNA. And therefore, it would be a mess, right? You would get a mismatch of proteins and stuff you don't want in the cell. Hopefully that makes sense. If you need, that's quite a complex topic. So if you need some clarification, make sure to ask, and I can respond in the comments or even make another video on this. So now the images are electron micrographs. So we have eukaryotic and prokaryotic DNA. So leading on from the last question, now it's asking you to distinguish between the structure of DNA in both. So in terms of the structure, well, think about it. This is quite simple. I think you can literally see it as a hint, but 3, 2, and 1. Well, first of all, in prokaryotes, you can see it's circular, right? It's a circular DNA loop. In eukaryotes, it's linear. And then secondly, in eukaryotes, it's associated with histones, which are proteins that, remember, help fold the DNA to make sure it can fit in the nucleus, because we have a lot of DNA, right? And our nuclei are not that big. Whereas in prokaryotes, there's no histones. It's called naked DNA, right? So those are the two main things, and you can see the histones here, right? Over there. Those are the histones. So, perfect. And now the last question, and this is paper 2B, the longer answer questions, which this is not that long, but it's the only one for theme A in the specimen paper for standard level, and it's distinguished between the thermal properties of air and water as they relate to the habitat of animals. So what I said before applies here as well, right? Describe first the different thermal properties, and then talk about how they relate to the habitat of animals. And here you would expect to write a long paragraph for 4 marks. So take some time, write it, take a couple minutes, maybe 5 minutes, and then pause this and come back. So 3, 2, and 1. So we have all of this, right? First, for the properties. So water has a higher specific heat capacity. It requires more energy to gain or lose, to change temperature. Then also it provides a more stable thermal habitat. This makes sense, right? Since it takes a lot more energy to change its temperature, the temperature stays a lot more stable. And then it also has a lot more higher thermal conductivity. So like energy, thermal energy, passes through water much more easily than air. Therefore, more heat is transferred from organisms to water than air. Now, what's the significance of this? Well, a lot, right? Basically, water is a very good evaporative coolant, and air is not. This is why we sweat, right? Sweat is to cool our bodies, and it's basically water because it's a good evaporative coolant. That's one of the consequences. It's because it takes a very high amount of energy to evaporate it. So we use the energy from our inner heat to evaporate it, right, and cool ourselves. So that's one consequence, and then another consequence is that aquatic animals need a lot more body fat as insulation. Why? Because of this high transfer in thermal conductivity in water. Otherwise, those aquatic mammals would just lose all of their heat, right, to the water, just through diffusion. But the body fat allows to internalize it, because remember, animals need to keep internal body temperature. Well, some of them. So yeah, perfect. So this is the last question. So let me know if you had any questions in the comments. In the meantime, make sure to do more practice papers, maybe from the past IB biology ones. And next week, we'll come back for the higher level ones for theme A, and then we'll look at the next themes in the following weeks. So hopefully this was useful, and I'll see you in the next one.